So anyhow, our next speaker is Trevor Marshall, and he is currently in the United States the director of autoimmunity research in Thousand Oaks in California. Since he is an Australian, he also keeps one foot in the door in Australia, and he's adjunct professor in the Faculty of Health Sciences at Murdoch University, and that is in Western Australia. So I think we just went from the south to the west. And we're ready.
Right, but we're still in the Antipodes, so that's something. Although most of my work now is based in California with the Autoimmunity Research Foundation. So I'm going to talk about the human microbiome. Well, what is the human microbiome? This is a web page, or images off a web page, which was produced by the National Institutes of Health about 3 or 4 years ago when they kicked off the Human Microbiome project. And the goal of the human microbiome is to look for microbes in and around the human body by using the DNA sequencing technology which is available these days. So instead of trying to culture the microbes, and we know that only 2 to 5% of microbes are capable of being cultured, the DNA of the microbes was going to be used to identify just what was a commensal or a pathogen with Homo sapiens. And the data has started to come in, and what we've found is that in Homo sapiens there is in fact thousands of species that are living in and around the human body, whereas we have thought for so long that the human body is a sterile compartment. It is not sterile. It's not even close to being sterile, and I'll show you some of the science in a short time. I'm going to go first right to the quick and show you what we've actually done, and then hopefully that will make you a little bit more interested in the science and keep your attention later on. What we contributed was that we noticed back about a decade ago that uniquely in Homo sapiens, not in the great apes, but uniquely in Homo sapiens, intraphagocytic pathogens can persist by weakening innate immunity. But innate immunity, not adaptive, by disabling the transcription of a single key nuclear receptor, the VDR, type 1 nuclear receptor. Because in Homo sapiens, and only in Homo sapiens, one nuclear receptor, the VDR, expresses genes for TLR2, as well as the cathelicidin and beta defense and antimicrobial peptides. Actually, cathelicidin is a protein that's broken down into peptides with microbicidal action. And all of those are essential to the intracellular innate immune defences. So if you're a microbe that wants to persist inside the nucleated cells of the human body, it makes sense that you have evolved to knock out that receptor. And in fact, that is what we found has been happening, because we found a drug that was already approved that we could retarget by changing the dosing. We could retarget this to reactivate VDR gene expression, or certainly expression of genes by the VDR. And one of the first signs that we had been successful was this paper that was presented in Japan at the 26th conference of the Japanese Society of Sarcoidosis and Other Granulomatous Disorders in 2006. And this was presented by Keisuke Arasaki. And Arasaki-san was reporting on the case of systemic sarcoidosis treated according to the Marshall protocol. I didn't choose that name, I'm sorry, but the therapy that we've developed was being used by Arasaki-san on a very sick patient in Japan with neurosarcoidosis and extremely disabled. And he was reporting very good results. And in fact that patient is now back at work practicing as a lawyer. It's a matter of interest. Then by 2008 we'd collected some more data. We did a quick retrospective analysis of it, and we presented at the International Congress on Autoimmunity in Portugal in 2008 about 100 patients with various diagnoses that were being treated with this VDR agonist. And a whole range of chronic diseases were found to be responding extremely well. We have a very unusual way of presenting it, this data, because we have a very low rate of failure. In fact, the rate of failure is only around 3 or 4 per 100 on this chart. So we've actually spelt out year by year, up until the 3rd and 4th years, how the patients responded to the therapy. Rheumatoid arthritis and Hashimoto's thyroiditis, of course, are right at the top. Uveitis, psoriasis, multiple sclerosis down here, psoriatic arthritis, diabetes, and then we just had a few patients with, well, one patient in fact, with myasthenia gravis and diabetes insipidus diagnosis. But by an average of 36 months into treatment, 81% of the cohort were reporting reduced disease and symptoms, and many of them have gone on beyond that point. This is one of the MS patient case histories, which was also presented in Porto separately by Dr. Greg Blaney from Vancouver in Canada, one of our worldwide collaborators. And it is tracing the case of a patient with multiple sclerosis, starting off with an EDSS of 8.5. You actually can't see that for some reason. Well, it doesn't matter. Believe me, the EDSS started at 8.5. It got to 8.5, 8.0, 7.5, 7.5, and 7.0 over a period of 4 years, roughly from 2006 to 2010. The patient was a 56-year-old female. The patient is a 56-year-old, 58 probably now, year-old female, diagnosed with relapsing remitting in 1995, progressed to EDSS 8.5 by September 2006 with paralysis in both legs and pelvis, incontinence and refractory to treatment. The VDR agonist olmesartan was commenced in March 2007. By June 2007, the lower spasticity had moderated, and by March 2008 it had dropped to mild. By January 2010, patient could walk 15 to 20 steps with assistance, contract quadriceps and hamstrings against resistance, had improved sleep, depression was minimal, and no longer needed diapers. The incremental improvement continues. Quite an amazing case history, but it's not just multiple sclerosis. Dysregulated VDR has been documented in a number of conditions: depression, multiple sclerosis, arthritis, lupus, sarcoidosis, thyroiditis, diabetes, dementia, autism, schizophrenia, and tuberculosis. This is from a paper by Greg Blaney, who featured in the earlier slide, which was in the Annals of the New York Academy of Sciences a year or two ago. So if we now start looking at science and look at this drug, you've got a receptor, and you've got a drug, a ligand, which sits in the binding pocket. In this case, it's olmesartan. And we have ways of studying to show that it's an agonist, but the way that I used was a very, very strong methodology. We used molecular dynamics. We actually simulated the operation of the VDR. Why did it stop? Probably stopped because I hit the mouse pad. It'll come again. We simulated the operation of the VDR. In real time, and with the ligand, with the drug in the binding pocket. This is the VDR. All molecules move at all times, usually fairly vigorously. It's a fairly large molecule. There are about 200 amino acids shown on screen there, and 4 or 5 major helices. But we were also able to do this using Rattus norvegicus, the rat VDR, because there was an X-ray model available for the rat VDR. And what we found was the drug doesn't work in a rat, which is probably why nobody else had noticed that it could be retargeted. In fact, if you look at the tetrazole ring here, there are 2 strong hydrogen bonds in the human, whereas in the rat the tetrazole is not hydrogen bonded. It does not create the forces necessary to activate the VDR receptor. And in fact, a rat or a mouse has different immune defences as well. This is a press release came out just 22nd of May. Ulcer bacteria may contribute to the development of Parkinson's, where they were able to induce Parkinson's into a mouse strain of middle-aged mice with a delay of about 3 to 5 months. But I want to caution that the immune system in a mouse is totally different. Probably need a totally different species than what is needed in Homo sapiens. Well, there are always problems. And there is a huge problem when you start to stimulate the immune system. And that is immunopathology. All of our bodies are carrying around very large loads of microbes. Most of those microbes are not pathogenic. They do not cause diagnosable disease. The Microbiome project, you may have seen on the first slide, has now estimated that about 90% of the cells... Excuse me, why are you moving that? Excuse me?
Sorry, it's yours?
Yes, that's mine.
Oh, I'm sorry.
90% of the cells in the human body... 90% of the cells in the human body are bacterial in nature. Now, admittedly, bacterial cells are very, very small, so the volumetric analysis would not be 90%, but by numerical analysis, about 90%. And when the immune system is enabled to start killing the pathogens inside the cells, then many of the cells themselves die by autophagy and apoptosis. And that necessarily increases the inflammation. So the inflammation, the level of inflammation, be it subclinical or clinical, that the individual was experiencing, necessarily increases when you start killing off the microbes. And if too many cells die at once, then the patient can become very, very ill. So immunostimulatory therapy has to balance the rate of healing against the amount of immunopathology that's going to be endured by the patient. There are many groups that have tied inflammation to neurodegenerative diseases. Fred Gage's group down at Salk Institute is the one that I've been following most frequently. But the thing that we all fail to realise, at least as far as I know, we all fail to realise is that once you start dealing with the source of the inflammation, rather than suppressing the inflammation, actually trying to allow the body to deal with the microbes at the heart of the inflammation, then you get immunopathology and you get a surge in disease symptoms. And here are some of the graphs from our most recent paper, which was immunostimulation in the era of the metagenome in cellular and molecular immunology, just January 2011. And the first one is from a rheumatoid arthritis patient, and we have the ANA titer versus time, 2,000 days on therapy. And initially the ANA titer was 1 to 160. It doubled after about a year and then gradually fell away. But the initial response is an increase in autophagy, an increase in apoptosis, increase in cell death, increase in disease symptoms. Similarly over here for an ankylosing spondylitis patient, the Bath ankylosing spondylitis disease activity index, actually that remained fairly stable and then started falling away. But the sedimentation rate rose and the C-reactive protein rose, peaking at about the 800-day or 2-year level roughly into therapy. And finally we have a sarcoidosis patient where we've plotted BUN, blood urea nitrogen, and glomerular filtration rate and creatinine against the number of days up to 2100, about 6-7 years. And you can see that they fluctuate, but what you probably can't see from the back is if you look at the GFR started at 70, which is not really good, but it started at 70 and fairly quickly by about 3 years it had dropped down to 40, and it's maintained stably at 40 for the subsequent 3 or 4 years of therapy. When you have a patient with a GFR of 40, you would normally be pressing the panic buttons. But when you're changing the paradigm of the therapy that you're using, the panic buttons change. It's a very difficult problem that we have to solve. So what do these pathogens look like? These are from a study by the Wirosco group. It's a transmission electron microscopy study in 1989. It was done at Columbia University in the US. And this is a lymphocyte from the eye of a juvenile rheumatoid arthritis patient, but they did a number of other patients as well. And you can see that there is a nuclear staining here for bacterial DNA, which clearly has not been properly phagocytized, and there are also longer artifacts as well, biofilm-like artifacts, that stain up quite nicely. You can see them better if you look up the original article by the Wiroscos. Under light microscopy, what you see is the occasional cell, about 1 in 100 monocytes, typically in a very ill patient, are infected. And this one, the cytoplasm, is heavily infected of a monocyte, and it's already starting to throw out these long polymers to try and spread the infection to other cells that are still relatively potent. You can see it with optical microscopy. It's very difficult. You're right at the limits of microscopy, and you really can't see as much detail as we would like to see to understand exactly how these pathogens survive. But the DNA analysis gives us a lot more information. Here is the salivary microbiome, which was produced by the Max Planck Institute as part of the Human Microbiome project, and the size of the text is roughly proportional to the percentage of that particular species or phyla that they found. This is very small, but on the other hand, it's Yersinia. Yersinia, which, as Yersinia pestis, one of the subspecies, was responsible for the Black Death. Yersinia was found in the saliva of healthy human beings from 12 different places on the planet, along with Neisseria, strep, and a number of other Enterobacter, a number of other organisms that we would regard as being pathogenic. And yet very few of these could actually be cultured. Some were cultured, very few could actually be cultured. Because when the microbes move into the microbiome, there is a lot of sharing of genome. Actually, genes, and especially plasmids, disappear. And you're dealing with a community of microbes rather than individual infestations of microbes. And this community builds up gradually during life. So where do the microbes come from? Everywhere. Here is the cigarette microbiome, which was published last year, I think, yes, 2010. And it shows that when somebody smokes a cigarette, they take in a very large load of Pseudomonas, Acinetobacter, Klebsiella, Clostridium, Bacillus, Proteus, and Staph down here at the bottom at about the 5% level. And there are other species, of course, as well that couldn't be identified or that are smaller than the 5% level. Think about it. The other thing that's very interesting is that the initial data from the COPD lung found that Pseudomonas is very strong in the COPD lung. The microbiome work is opening an entire new insight into how the human body functions, and in particular how it dysfunctions. So some of you will say, oh, it's contamination. So here, let's go to an internal sample rather than external samples, saliva and skin and things. This is these are the species that were shaken off a hip joint that was replaced with revision arthroplasty. A number of species, Lysobacter, it was a biofilm that had collected on the old joint, so you'd expect Lysobacter to be there. But look, this one I want to point out, hydrothermal vent eubacterium. a bacteria that had been previously identified in hydrothermal vents at the bottom of the ocean. And here was their DNA turning up inside a human being. That's not the sort of bacterium that you typically find floating around a surgery as a contaminant. Some of the bacteria that we already know downregulate the VDR as their survival mechanism. Epstein-Barr virus, which of course is associated with just about every inflammatory disease. You'll find something in PubMed pointing at EBV. But if you look here at the VDR, you'll see that it's very heavily downregulated in the lymphoblastoid cell lines. After 1.5 years, expression of VDR has been attenuated down to extremely low levels. And even after 2 months, it's well attenuated as well. Whereas in the peripheral blood cells, there's not that much activity by the EBV. That's not surprising. EBV is a persistent, certainly a very persistent pathogen. Also, Mycobacterium tuberculosis. Some very good work done here in China in 2003, which showed that the VDR receptor was downregulated by when a cell was infected with Mycobacterium tuberculosis. And similarly, Borrelia burgdorferi, very heavily live BB 50-fold downregulation of VDR, a very, very strong one indeed. But if you look at species that you don't really regard as pathogens, this is a Manhattan plot of the genomes of Fusobacter and the human chromosomes, the 22 human chromosomes. And what it shows is the degree of homology, the degree of similarity between the genome of the bacterium and the human genome. And you can see some of these similarity points are really, really high. And there's very likely to be interference between the proteome and the genomes of these pathogens when they become part of the microbiome. And Fusobacter is a very common constituent of the microbiome. So what we have to say is that the catastrophic failure of the human metabolism, which we see in chronic disease, which at first glance appears so diverse, hundreds of separate diagnoses for typical chronic inflammatory diseases, is actually due to a single underlying mechanism, which is a microbiota which has evolved to persist in the cytoplasm of nucleated cells by dysregulating the VDR nuclear receptor. And interference of the both genomic and proteomic functions of the cell takes place on a huge scale with imponderable complexity, leading to a semi-infinite number of subsequent disease syndromes. In other words, if you've got 50 or 600 it was in this library microbiome, but let's just take 50 genomes that are in the cell and persisting in the cell, each of them holding around between 500 and 5,000 genes, and then each of those can interact with the 25,000 human genes, the siRNAs, all of the other internal cell operations. And remember, everything happens in the cell. It doesn't happen at the level of the tissue. Everything happens in the cell. All the proteins that the human body needs to operate come from the cell. We've got to focus our thinking down from the level of the tissue to the level of the cell. So in closing thoughts, you would have noticed that there was relatively little data. We had good data on rheumatoid arthritis and thyroiditis back there. We also have good data on neurosarcoidosis, depression, particularly manic depression, bipolar disorder, chronic fatigue syndrome, and many autoimmune disorders to be quite happy with the proof of concept. But we're still accumulating more long-term recovery data in multiple sclerosis, schizophrenia, OCD, dementia, multiple system atrophy, and amyotrophic lateral sclerosis, all of which are responding well, but in very small numbers at this point. The other thought I want to leave you with is the thought that the previous speaker had, which is that by the time the brain becomes infected, many systemic pathways are also already active, and vice versa. by the time the systemic disease becomes manifest, the brain is very often affected, with a few exceptions like ALS that I'm obviously intrigued with. As we move to the future, there really has to be a choice made by the physician or by the patient between immunosuppression to give short-term productivity, long-term relapse and morbidity, and immunostimulation to give short-term disability and a long-term productivity. The World Health Organization has just declared a chronic disease alert, saying that death from non-communicable disease is on the rise, with the developing world hit hardest. I've got news for them. Non-communicable is arguable at this point in time. And if you want a book to read what's happening, the textbook has just been written on metagenomics of the human body, which is edited by Karen Nelson, who heads up the Maryland campus of the J. Craig Venter organisation. We wrote the chapter in there on autoimmunity. It's a Springer textbook, and it's one that I'd certainly recommend you pull from the library and read. Thank you very much.
Questions? Yes.
Well, thanks so much, Trevor. It's absolutely mind-boggling stuff. And what could we look for, though? I mean, like you say, these bugs, if you'll pardon the phrase, working within the cells. So there's obviously no sort of external tissue-level signs of normal septic processes, apparently, as I understand it.
Right.
So how could we test for their presence?
Well, it's even more complex than that.
Right.
That optical microscopy that I showed you. What we do there is we put blood between two cover slips and then allow the blood to age. And between about 6 and 36 hours, the cells that are infected start to explode like that. So you look at them after 36 hours and there is just debris. There's no signs of cells. So in typical sampling operations that one does on a clinical basis, you don't see this.
I mean, we have access to regular microscope slides of human pathological brain tissue, for example. And, if you're looking at a Parkinson's brain,
yes,
you can't see anything.
But you can sequence it. But you can sequence it and you can send it to the Argonne National Lab metagenomics analysis. You just upload your raw data and it compares the RNA that it finds, not just against the human genome, but against the 6,000 odd complete genomes that are available out there for microbes.
What I'm trying to understand is if these little bugs are actually causing diseases, like these processes, then there would have to be something you could see if you look carefully at individual brain cells, for example, some sign of what is developing.
Yes. There was an image that I got from Karolinska when I was there of a Parkinson brain. You see the cell, and under backlight illumination, you see usually bright areas, the proteins that protect the colonies. If you remember, there was that vacuole type colony. They tend to come up under backlight illumination. And you can just see the blobs there if you use the highest magnification of your optical microscope. And it's about 1 in 100 cells that are infected, so you've got to look a fair bit. It's only the nucleated cells. You've got to reject all of the red cells. But then you can see them, yes.
I have another question. The patients you showed, you followed for years.
We've followed right through from 2002 through to today, most of them. We've lost some to follow up, but we've followed everyone that we can.
And your efficacious time seems to be 1 to 2 years, and then you have follow-on.
Response time is one. Well, there's the initial response of immunopathology, which is instant, a month or less. And then there's the initial turning the corner, light at the end of the tunnel, which is 1.5 to 2.5 years, typical. I think we said by 36 months, 81% have got to that point. And then there is the recovery period. And depending on how ill they are, they become functional again. They get back to work, get back to the community at some point beyond that.
Right. So if you design a study for the FDA or to get approval for this drug for your indications of all the chronic degenerative indications you have mentioned, how would you design the study and how long would it be?
Well, we're discussing that with the FDA at the moment. The problem is that FDA doesn't like any studies going over about 18 months.
They have a lot of follow-ons.
Right. But in terms of the phase 2 studies for the NDA, they like them to be closed-ended. And we would really not be getting results until a longer term. We're trying to persuade them at the moment that immunopathology is an endpoint, which in and of itself is important. And in time we'll be able to do that, I think. How quickly, I have no idea.
Thanks very much. Yep.
And incidentally, the drug that we're putting through the FDA is a purified form of the drug that's already on the market. So the drug is already available on the market. Sorry about that.
I'm sorry, I'm interrupting and hurrying up in between. Very short question. Olmesartan, which you have advocated, which can inhibit the VDR gene, how does it actually work in this autoimmunity? This one. And the dysregulation of VDR gene has been found in multiple sclerosis but not in motor neuron disease. Is it just because we have not tried to identify this dysregulation in motor neuron disease?
I would want to talk to you about that later and have a look at the motor neuron disease, because I frankly haven't looked at it in motor neuron.
Can you just briefly explain the mechanism with which this whole set of symptoms are brought back by olmesartan?
Very quickly, and I don't mean very quickly in answer, but very quickly the body changes, the hormonal balance changes very quickly within 2 to 3 weeks. If people are on thyroid medication, very quick, that has to be continuously modified, because usually within 18 months they're off thyroid medication. The whole balance changes very quickly with the immunopathology setting in and with the therapy. But look, it's probably better if we deal with that offline, because I've run over time and I don't want to take time from the other speakers.